Welcome, everyone, to this webinar event. My name is Jose. I'm part of the scientific support team here at Twist, and I will be your host today. Before I introduce our speaker, I would like to remind our attendees of a few housekeeping items. All lines will be muted during the webinar. We will answer all of your questions after the presentation. And if you have any questions, please submit them in the Q&A box rather than the chat box. And finally, following the presentation, if you have 1 minute to take a brief survey, we would really appreciate your feedback. And with that out of the way, I have the pleasure to introduce our speaker. Prashanth Srinivasan is a bioengineer based in California in the US. He currently works as a scientist and project manager at Antheia, a synthetic biology startup, developing microbial platforms for mass-producing plant-derived medicine. Born and raised in Canada, Prashanth received a bachelor's degree in biochemistry and chemical engineering at the University of Ottawa in 2016, and completed his PhD in bioengineering in Dr. Christina Smolke's lab at Stanford University in 2021. During his PhD, Prashanth developed a new cellular engineering technology to convert brewer's yeast into a microbial factory for Tropane alkaloid drugs. In his spare time, Prashanth enjoys growing medicinal and often poisonous plants. And with that said, off to you, Prashanth.
Wonderful. Thank you so much for the introduction. Welcome, everyone. I'm delighted to have this opportunity today to share with you all some highlights from the research that I conducted during my graduate work at Stanford, specifically on engineering yeast cellular factories for more sustainable production of plant-derived medicines. So in this talk today, I'll be using a series of case studies from my own work to try to illustrate how rapid advances in DNA sequencing and synthesis technologies are not only enabling the type of work that I've done to be conducted much faster than ever before, but they're also driving an important paradigm shift in the field of metabolic engineering. So I will start by providing a little bit of background on what motivated me, and I imagine many other researchers in this field, to pursue this idea of developing alternate supply chains for plant-derived drugs. When you hear the phrase plant-based medicine, a lot of people tend to think of herbal remedies or traditional medicines maybe found in pre-industrial cultures. But it might surprise you to learn that modern healthcare systems, even those in so-called developed countries, are still heavily reliant on plants as a source of pharmaceuticals. So by recent estimates, about 70% of the global population still relies on plant-derived products for their primary healthcare. And if you were to take an inventory of all the drugs found on the World Health Organization's list of essential medicines, you'd find that up to 40% of them are either extracted directly from plants or are made from plant-derived precursors at some stage. And that's because no other domain of life comes even close to the diversity and variety of bioactive molecules that plants are able to synthesize. Fortunately, many of which have medicinal properties for humans. So shown on this slide is just a small subset of some of the plant-derived pharmaceuticals and drugs that are in widespread use today around the world. So, for example, the anti-malarial drug artemisinin, which is extracted from the wormwood plant, the very widely prescribed painkillers morphine and codeine from opium poppies, a compound called paclitaxel, which is a powerful chemotherapeutic, an anti-cancer agent from the Pacific yew tree, colchicine, which is a very widely prescribed anti-inflammatory for treating things like gout from the flame lily, some more important anti-cancer drugs from the Madagascar periwinkle, and of course some exciting and very potent new psychotherapeutic drugs from cannabis. Now, another group of plant-derived drugs with very widespread and worldwide use, and which were the focus of my own graduate work, are the Tropane alkaloids, or TAs, as I will be referring to them. These molecules are produced by several different plant families, including most famously by the nightshades, or the Solanaceae, which is actually a family that includes food crops like tomatoes, potatoes, and peppers. But they're also made by other plant families, including potentially more infamously, in the example of cocaine, in the coca family. But TAs produced by certain nightshade plants, which are pictured here, are used for treating very diverse neuromuscular diseases, ranging from gastrointestinal spasms, cardiac muscle problems, and cardiac failure, all the way to nerve agent poisoning, and even more recently, Parkinson's disease. And perhaps most recently, tropane alkaloids have been used to facilitate intubation when placing patients on ventilators in advanced stages of COVID-19 and other respiratory diseases, a use that, as a result of the pandemic, has actually contributed to ongoing shortages for some of these drugs. Now, global production of tropane alkaloid drugs depends on intensive cultivation of Du Boisia trees on plantations like the one pictured here, mainly in eastern Australia. And so far, no efficient total chemical syntheses for these molecules have been reported. We continue to have to rely on the cultivation of medicinal crops on plantations like this one to mass produce these drugs. But unfortunately, increased demand for essential medicines around the world over the past century or so has made it very clear to us that current plant-based supply chains for many, if not all, plant-derived drugs are simply not sustainable. And the Tropane alkaloids are a perfect example of this vulnerability, as there are several critical weaknesses associated with this supply chain. Now, first is the issue of geographical restriction. Now, because the majority of global production of these drugs is still concentrated in Eastern Australia, this means that any environmental perturbations or disasters that affect one location can severely impair the entire global supply. And we saw this, for example, at the end of 2019, the start of 2020, when a series of devastating wildfires raged through nearly the exact parts of Eastern Australia where many of these large Duboisier plantations are located. And given the accelerating pace of climate change, these kinds of disasters are only going to increase in frequency and severity. There is also, of course, the issue of environmental degradation caused by plant-based supply chains. So Duboisier plantations, for example, require the clearing of large areas of native ecosystem, as well as huge inputs of irrigation and fertilizer, all of which have very long-lasting negative impacts on the surrounding ecosystems. And the third major weakness with these plant or crop-based supply chains was really highlighted by the ongoing, or hopefully almost over, COVID-19 pandemic that I mentioned, as well as shortages for drugs like atropine, which were in high demand. With a crop or plant-based supply chain, especially one that relies on slow-growing trees, there's really no easy way to quickly scale up production in response to changes in demand or spikes in demand. Fortunately, plants aren't the only way to make plant-derived drugs. We can actually transplant an entire biochemical pathway, for example, a plant pathway to make tropane alkaloids, from plants into an entirely different organism. And for example, we can use organisms like microbes as the host for this transplanted pathway, which then allows these microbes to start making these same molecules. And yeast, the same microbes that have been used by humans for millennia to make alcohol and bread, is a particularly useful platform for making plant-derived drugs. So not only has yeast been extensively studied with a wide variety of tools for engineering, but yeast cells also have a similar architecture as plant cells, which helps to minimize the chance of any cross-platform compatibility issues that might arise when you transplant a biochemical pathway from one kingdom of life to another. Yeast are also quite comfortable growing in large vats or bioreactors, meaning that there are very well-established methods for scaling production to industrial levels. And by brewing drugs in closed bioreactors, you're making them much less susceptible to environmental disturbances with a much smaller land and resource footprint than growing plantations of medicinal crops. So for all of these reasons, the objective of my graduate work was to try to engineer yeast, the same kind of brewing yeast that's used for alcohol and bread, to convert simple building blocks like sugar and amino acids into the two most commonly prescribed tropane alkaloid drugs, hyoscyamine or atropine and scopolamine. But as I would soon discover, although microbial platforms can avoid a lot of the weaknesses of plant-based supply chains like I was talking about, the process of actually building a microbial platform in the first place is still an enormous engineering challenge. And because each pathway reconstruction task is so unique with its own set of novel problems and failure points, what I've learned is that there is very limited capacity to transfer learnings from one pathway to another. So during my PhD defense, one of my committee members actually asked me which technology I felt was the most critical to the success of my work and to modern metabolic engineering. And my answer was DNA sequencing and synthesis technologies. So let me try to provide an example to illustrate why I came to this answer. In our lab, that is the Smoky Lab's previous work at Stanford, it took a team of over 6 very talented scientists and students almost 10 years to develop a proof of concept yeast production platform for making pain-killing opioids like thebaine and hydrocodone. Now, overall, this engineered pathway involved a little over 20 additional genes put into yeast and many tens of person years to complete. But rapid advancements in the availability of DNA sequencing datasets and reductions in the cost of gene synthesis over just the past 10 years have changed things quite dramatically because they enabled a microbial production platform for tropane alkaloids, which I'll be talking about today, which was also at the time the most complex pathway ever reconstructed, to be completed by just 2 people in under 5 years. Now, this is in no way an indictment, obviously, of the abilities of the previous scientists. If anything, they're all far more talented researchers than I am. But instead, this comparison is intended to really highlight the immense impact that advances in DNA sequencing and synthesis have had in accelerating the pace of metabolic engineering. But in addition to improvements in the speed of research, these advances are also driving a very important paradigm shift in metabolic engineering, away from the kind of traditional bag of enzymes model, where you think of cells simply as self-replicating containers of chemical reactions, and towards a more integrated, multidisciplinary approach that is driven by DNA sequencing and synthesis. So over the course of this talk, I'll try to draw on some specific examples from my graduate work to illustrate 4 specific avenues along which I've observed this paradigm shift occurring. So in the first case study I'll talk about today, I'll discuss how previously pathway discovery in native host organisms and pathway reconstruction in heterologous hosts were 2 very separate tasks performed by 2 different teams and requiring different organisms and very unique skill sets. But with the public availability of genome and transcriptome data sets now increasing, this has enabled these tasks to be done synergistically using microbes to accelerate the design, build, test cycle. In the second case study, I'll describe how rapid gene synthesis allows us to act not only as metabolic engineers, but as cellular engineers. So not just modifying metabolic pathways and their associated enzymes, but also modifying how these components interact with fundamental cellular processes, like, for example, protein trafficking pathways. In the third case study, I'll illustrate how machine learning tools can be combined with sequencing data sets to optimize the compartmentalization and spatial organization of engineered biochemical pathways and their metabolites. And in the final, very brief case study, I'll describe how some newly developed chemoinformatic tools and some, I guess what I'll call non-traditional decision-making criteria, like ecology and ecological relationships between organisms, can actually act as very powerful tools for informing metabolic pathway design. So I'll start with the first case study, which was combining pathway discovery and pathway construction. So I won't dive too deeply into the details of this Tropane biosynthetic pathway, as the steps in their individual details are covered in the associated papers, but instead I'll use this opportunity to provide a very high-level tour of the pathway, as well as, more importantly, the major challenges that we faced in assembling it in our yeast platform. So this pathway, which is shown in the schematic on the right side, is divided into 5 metabolic modules. Modules 1 and 2, which are highlighted here, enable the conversion of this starting molecule, an amino acid called arginine, into this first linear intermediate called putrescine, by way of the urea cycle and polyamine metabolism. And then through a series of ring-forming or cyclization reactions to form this bicyclic intermediate called tropane, which is shown at the bottom of module 2, sort of in the center of the schematic. Tropane comprises 1/2 of the full tropane alkaloid skeleton. Now, the major challenges we faced in these 2 modules involved figuring out, first of all, how to break the tight feedback control that yeast uses to restrict polyamine accumulation, as well as how to promote the desirable non-enzymatic or spontaneous steps in this pathway, while at the same time limiting the spontaneous undesirable side reactions. But in general, these are pretty typical challenges that you'll face in a metabolic engineering project. The second half of the tropane alkaloid skeleton comes from a second amino acid called phenylalanine, which is shown in the sort of top right of the schematic there. And this amino acid is converted through 3 steps in module 3 into a reactive intermediate molecule called phenolactic acid, or PLA glucoside. That's shown towards the bottom of that module. And the primary challenge we faced in module 3 was finding a phenylpyruvate reductase enzyme, that's indicated in red, the PPR, that was actually active in yeast, because many of the bacterial and plant enzymes that we tested were completely non-functional. Module 4, which is highlighted at the bottom of the schematic now, comprises a series of downstream rearrangement and oxidation reactions to convert the full tropane skeleton into our medicinal target compounds, hyoscyamine and scopolamine. And finally, module 5, which is highlighted in this sort of pink compartment here, comprises the central esterification or acyltransferase reaction, in which the 2 halves of the tropane skeleton, tropene and PLA glucoside, are condensed together. Now, modules 4 and 5 really posed much more significant engineering challenges. The wild-type acyltransferase enzyme that's currently shown as being central to module 5 was initially, in our hands, completely non-functional in yeast, and we had no idea why. And in module 4, which is highlighted at the bottom, no known enzyme existed in nature to catalyze the reduction of hyoscyamine aldehyde, which is this intermediate here, to hyoscyamine, meaning that an enzyme discovery project was needed. And so this enzyme discovery project is really the focus of my first case study. So the traditional approach to discovering missing enzymes in plant biosynthetic pathways has a very high success rate, But it's also very time-consuming and requires a lot of specialized skill. And for more details on this process, I'll direct you to this very excellent and thorough mini-review that's cited in the bottom corner of the slide there. But I'll try to briefly summarize this process just for comparison. The first major task is identifying a set of candidate genes for your missing enzyme. This typically involves metabolic profiling experiments in the native plants that possess the biosynthetic pathway or that make the molecule that you're interested in, as well as some comparative pathway analysis between related plant species. Once a probable enzyme class has been identified, a set of candidate genes that match this class can be identified either from a whole genome or a whole transcriptome data set for that plant. But obviously, depending on the enzyme class, this initial set can be prohibitively large for testing. In the case of a very highly represented enzyme class like a cytochrome P450 or an oxidase, you can have on your hands many hundreds of potential gene candidates for testing. And so the second major task is to try to reduce the initial candidate set to a much higher likelihood subset that is much more practical to test. And this is often done using a combination of computational tools like comparative homology, as well as direct plant experiments like random mutagenesis with metabolic profiling, or treating plants with specific stimuli or elicitors and then performing gene co-expression analysis. But obviously, any approaches here that depend on the generation of plant mutant lines, or even just growing plants in general, can take several weeks to months to complete. In any case, once a smaller candidate subset is generated, the actual missing gene can be identified, hopefully, and validated either through gene knockout studies in the native plant and or through expression in E. coli, purification, and in vitro assays. Now, overall, the point I'm trying to make by summarizing this pipeline is really that the most laborious and lengthy steps in this discovery pipeline really depend on growing plants, which unfortunately can often take a long time. But fortunately, eukaryotic microbial testing platforms like yeast can be used to significantly speed up this pipeline through candidate library screening. So for comparison, on this slide, I'm going to show you a typical workflow and timeline that we'd use for a design build test cycle, or a DBT cycle, if you were to use yeast as your screening platform for gene discovery. So we can go from receiving and then cloning your new genes of interest, your candidate genes, into expression vectors, transforming them into yeast, growing cells, and analyzing their molecular production in under 2 weeks, or really under a month if you're including the time needed for gene synthesis. And because of how easy it is to transform and grow yeast, you can complete several DBT cycles or several rounds of candidate screening in the time that it would take to complete just one round with the traditional plant-based workflow. And so given our lab's previous expertise with yeast, as well as the significant time advantage that's promised by this platform, we decided to apply a yeast screening approach to try to discover that missing oxidoreductase enzyme that I mentioned existing in module 4 of our Tropane pathway. So in this downstream module 4, which is again highlighted in the schematic on the right side of the slide, 2 out of the 3 enzymes that we needed were already known. A cytochrome P450 called CYP80F1 catalyzes this internal rearrangement of liturine, which is the initial product of tropane and phenolactate glucoside, those two halves coming together into hyoscyamine aldehyde. And then a bifunctional 2-oxoglutarate-dependent oxygenase called H6H catalyzes this 2-step epoxidation of hyoscyamine to scopolamine. But as indicated by that question mark in the middle, to discover that missing enzyme catalyzing that middle step, we applied an unsupervised machine learning approach using linear clustering methods and a publicly available genomics database for deadly nightshade, which is a tropane-producing plant shown on the left there. And we use this approach to try to identify any oxidoreductase candidates that show similar patterns of expression as known tropane pathway genes across different plant tissues. And we specifically focused on secondary root tissues because these are the tissues in plants where tropane alkaloids are actually synthesized. And so a graphical representation of this clustering analysis is shown on this heat map in the middle of the slide, with the candidate genes listed along the y-axis vertically, and then their expression shown as color intensity, with green being very high expression in the heat map, and it's shown across different tissues which are listed across the x-axis. So you can see flowers, mature seeds, different kinds of roots, stem, fruit tissues, etc. And you can see that most of our candidates, which are listed in blue and black there, show the same highly root-specific expression as known tropane genes, which are shown in purple. And you can see there's this strong overexpression along the secondary root column. So based on this type of co-expression analysis, we were able to reduce our candidate set from initially hundreds of potential oxidoreductase candidates to just 12 complete sequences with similar expression patterns as known pathway genes. And we then tested this entire candidate set in a single round using our accelerated yeast screening platform that I talked about a few slides ago. And one of the 12 candidates that we tested, which we named hyoscyamine dehydrogenase, exhibited the missing reductase activity that we were looking for and able to convert hyoscyamine aldehyde to hyoscyamine. And then using a variety of phylogenetic, structural, and mutagenesis analyses, we showed that this novel reductase, whose homology model is shown here on the left, is a zinc and NADPH-dependent member of the medium-chain dehydrogenase family, and potentially more interestingly, could have evolved from an ancestral enzyme involved in lignin biosynthesis. But with the discovery of this final missing step, we were able to complete this downstream module 4 for converting littering to scopolamine. And overall, as a result of this accelerated yeast pipeline that I've been talking about, the whole process from accessing the public transcriptome to identifying the missing candidate took less than a month, which is orders of magnitude faster than the traditional plant gene discovery pipeline. So in that first example, I showed how the public availability of genome and transcriptome data sets can be coupled with a microbial testing platform like yeast in order to cut down the time required for gene discovery to just a fraction of what would normally be required for a plant-based workflow. And so in the second case study, I'll use as an example the very next engineering challenge that we faced in this project, that central vacuolar acyltransferase step, to demonstrate how rapid gene synthesis can also speed up how we troubleshoot problems when proteins don't express properly in heterologous hosts. So the root of all the problems that we experienced with this central acyltransferase step, which is highlighted here in that pink compartment, all originated from the fact that in plants, this type of reaction typically takes place inside the plant vacuole. In fact, plants very frequently make use of their cell vacuoles as a sort of part-time biosynthetic compartment. In yeast, however, the vacuole's primary function is to break down and recycle enzymes and proteins, which means that the protein trafficking machinery that's needed to fold and transport a foreign enzyme to the vacuole is simply not present in yeast. Now, in nightshade plants, the two halves of the tropane alkaloid skeleton that I mentioned, tropene and PLA glucoside, are joined together by an acyltransferase enzyme called littering synthase. Now, unlike most plant acyltransferases, which are expressed without any sort of modifications, very simply in the cell cytosol, littering synthase moves through a rather complex protein trafficking pathway, which is shown on the schematic on the left side. And along this trafficking pathway, it receives various post-translational modifications, including disulfide bonds and proteolytic processing in the endoplasmic reticulum, as well as glycosylation of asparagine residues, which is called N-glycosylation, in the Golgi, before reaching its final destination, which is the vacuole. Now, this complex trafficking pathway in plants obviously poses a lot of problems when moving the enzyme into yeast. First of all, how many, if any, of these modifications are actually required for the activity of the enzyme? Does the enzyme need to be in the vacuole in order to be functional? And if so, how are the substrates that it's going to use going to enter the vacuole, and how is its product going to leave? And perhaps most concerningly, our initial attempts to express this littering synthase enzyme in yeast not only showed no activity, but also caused a major growth defect. So we tried really an exhaustive range of troubleshooting experiments across many, many different DBT cycles to try to identify what exactly was malfunctioning with this littering synthase enzyme, which fortunately we were able to complete in a surprisingly short time frame because of rapid gene synthesis. So we first suspected that the enzyme was not being expressed in the correct yeast compartment. I mentioned how in plants it's found in the vacuole, but perhaps its activity is best suited for the cytosol or maybe some other compartment. And so in order to alter the enzyme's location in the cell, we sourced a set of synthesized organelle localization tags and then loaded them onto the enzyme sequence to generate a sort of localization library. However, forcing the enzyme into every other yeast compartment using this localization library didn't yield any observable activity. And so our second hypothesis was that maybe the enzyme wasn't receiving the requisite post-translational N-glycosylations. Protein glycosylation patterns in plants and yeast are different, both in their recognition of specific residues to modify, as well as in the number and the type of sugar groups that are actually added to the protein. So to test this hypothesis, we synthesized a full combinatorial set of glycosylation site mutants. These are sequences where the specific residues that are modified are mutated so that they can't be modified. And we tested this, screened this glycosylation site library by comparing their sizes by Western blot in both yeast and in tobacco, which is another native model organism for nightshade plants. However, with this test, we saw that at least some fraction of the yeast enzyme matched the glycosylation patterns that were found in plants. which suggested to us that glycosylation was unlikely to be the primary issue causing a complete lack of activity in yeast. So our third hypothesis for why it might be malfunctioning concerned post-translational enzyme processing. I mentioned that some acyltransferases in this family are cleaved after translation in the ER in order to remove an internal linker sequence that results in a heterodimer that's held together by disulfide bonds. And so to test this theory, we compared the sizes and the activity of many different synthesized glycerine synthase linker variants that are shown here on the right side of the slide. And we did this using a sort of modular mix and match approach, including designs with split or separated domains, truncated or added back signal peptides, linkers from other acyltransferases that are known to be cleaved in eukaryotes, different artificial linkers, and unfortunately, none of these variants that we tested showed any activity. However, we did get a critical first clue into solving this mystery from this same Western blot result. The final linker variant that we tested contained this polyarginine sequence, that's shown by the sequence of Rs here in pink. And this polyarginine sequence should be recognized and cleaved by a native yeast protease called KEX2 that's found in the Golgi in yeast. But we observed, as you can see from this empty box here on the Western blot, that there was no processing of liturine synthase by KEX2, indicating that the enzyme wasn't even reaching the late Golgi. It was in fact getting stuck in this trafficking pathway somewhere earlier, sort of protein trafficking jam. And the second clue that we needed to solve this puzzle came from fluorescence microscopy. We observed that unlike the wild-type liturine synthase, which I mentioned causes a severe growth defect, the GFP-fused enzyme expresses without any issues in yeast and enables vacuolar localization, which you can see in these fluorescent micrographs on the top left. And so given that the vacuole is the default sorting location for transmembrane enzyme proteins in yeast, we suspected that maybe fusing green fluorescent protein, or GFP, to the N-terminus of this enzyme somehow turns it into a transmembrane protein, with the N-terminal signal peptide now acting as a central membrane anchor. And by testing a synthesized library of N-terminal fusion domains for liturine synthase, which are listed here and are shown here in this table, we actually saw that not only is the GFP-fused liturine synthase now active in yeast, but N-terminal domains with a greater propensity to oligomerize, like tetrameric DS-red, enable even higher levels of activity. And that's shown by this kind of progression going from monomeric to dimeric to tetrameric N-terminal domains. So this was a really exciting result because not only did it allow us to finally complete our reconstruction of the full tropane alkaloid pathway in yeast, but it also demonstrated how rapid gene synthesis, cellular engineering, and conventional metabolic pathway engineering strategies can be combined together to perform a sort of accelerated DBT cycle that is specifically tailored for troubleshooting protein expression and localization in heterologous hosts. But now that we had a functional acyltransferase enzyme in the vacuole, we were faced with yet another challenge. How can we ensure that the reactants and products of this enzyme are transported efficiently into and out of the vacuole? And so in this third case study, I will describe how supervised machine learning approaches like neural networks can be incorporated into traditional enzyme discovery pipelines to identify pathway-associated transporter proteins and how these transporter proteins can be used to support complex biosynthetic pathways that are distributed across multiple compartments. So plants have obviously evolved a diverse array of transporter proteins that they use to facilitate the movement of molecules through pathways that are distributed across organelles, cells, and tissues, many different levels of spatial organization. But when metabolic engineers transplant these pathways from plants into microbes, the associated transporters are usually left behind. Now, in this tropane pathway, there are 3 critical transport steps, and they're all located around the vacuole. And shown on the right side of this slide is a simplified schematic, a very high-level schematic, that just highlights the sort of transport events that are important in this pathway. The 2 substrates of the vacuolar liturine synthase enzyme, tropene, which is shown on the left side, and phenolactic acid glucoside, which is shown on the top, need to be transported from the cytosol into the vacuole lumen, and then the product of this reaction, liturine, needs to be exported back out to the cytosol. Now, transport of the glucoside into the vacuole is fairly straightforward. As an uncharged molecule, it has a relatively decent intrinsic membrane permeability, which means it can enter the vacuole through simple diffusion or maybe using some native sugar transporters. But tropene and liturine, unfortunately, are much trickier. Both of these are alkaloids and therefore are protonated on their nitrogen at physiological pH, and so their positive charge gives them very poor intrinsic membrane permeability. And this is especially the case for liturine, which is going to be entirely protonated in the acidic environment of the yeast vacuole, not to mention it's just a larger molecule. And so we tried to address these limitations by incorporating into our yeast platform plant vacuolar transporters to try to help with these molecular movement steps. So first of all, to facilitate vacuolar tropane import, that's import of the substrate into the vacuole, we expressed in our yeast platform 3 nicotine transporters from tobacco that were previously shown to also have transport activity on tropane, and those are shown here along the bottom of this graph. And 2 of these 3 transporters that we tested, JAT1 and MATE2, actually increased hyoscyamine and scopolamine production when expressed in our yeast platform. And fluorescence microscopy, shown on the bottom left of the slide, showed that these 2 transporters, as we suspected, localized to the vacuole membrane in yeast, indicating that they are most likely facilitating tropane import in our engineered strains. So with that improvement, we shifted our focus to the last of the vacuolar intermediates, littering. Unfortunately, unlike with tropane, no transporters for littering or any other fully assembled tropane alkaloids have yet been reported. But as tropane-producing plants likely have 1 or more transporters to facilitate this vacuolar littering export step, we decided to try to discover this missing transporter. Now, to do so, we initially used a similar sort of clustering and heat map approach, also referred to as unsupervised machine learning, to the one I showed earlier for identification of that missing oxidoreductase from the plant transcriptome dataset. Unfortunately, this approach proved ineffective here as it resulted in far too many transporter candidates to reasonably clone and test. In this case, well over 100 putative littering transporters. So we decided to test whether supervised machine learning methods like neural networks might be more efficient in searching for non-enzymatic pathway-associated genes like transporters. So starting with a publicly available transcriptome dataset for deadly nightshade, the same one I mentioned earlier, we split this dataset into 3 groups of genes according to this flowchart shown here on the left. First, genes that are known to be involved in the tropane pathway, so these are shown in green and indicated as TA genes. Second, genes known not to be involved in the pathway because they are either structural proteins, cell cycle regulators, or some other function that we can be reasonably confident isn't involved in biosynthesis. Those are shown in red at the bottom. And finally, in blue, genes that may potentially be involved in tropane alkaloid biosynthesis. So these include enzymes, transporters, and transcription factors, but without any prior characterization. Using the 2 definitive groups here, the known tropane genes and the known non-tropane genes, we trained 3 different binary classifier models to predict whether any particular gene that was presented to the model is or is not involved in tropane biosynthesis based on that gene's expression or abundance profile across 11 different plant tissues. So that's leaves, fruits, roots, stems, shoots, etc. And shown here are just the prediction results for the 3 models that we tested, which I'll just summarize by pointing out that of the 3 models that we tested, it was the neural network, which is pictured here at the bottom, that showed both the lowest false positive and false negative rates. So the way that this network works is a gene is presented to the network as an expression profile, that's basically expression levels across all these 11 different tissues, and what comes out of the network is a binary value, a 0 or a 1, indicating whether that gene is or is not likely to be involved in tropane biosynthesis. So the approximately 11,400 genes that we initially annotated as function unknown, that's the blue category, were presented to this trained neural network, and it predicted 33 new, potentially uncharacterized tropane-related genes, which are summarized in this table. And of these putative novel tropane-related genes, only 3 of them, which are highlighted here, were annotated as organic small molecule transporters: a lactose permease, a carbohydrate transporter, and a purine transporter. Now, obviously, paring down 30,000 candidate genes to just 3 is a significantly better reduction in search space compared to the more than 100 transporter candidates from the clustering and heat map analysis, which is still the standard approach for gene discovery in the field. But our data here is suggesting that some supervised machine learning methods, like neural networks, may actually be a more efficient option when looking for non-enzymatic pathway genes, like transporters or maybe even transcription factors. And so we next proceeded to test these 3 transporter candidates in our yeast tropane biosynthesis platform to determine if any of them were the missing vacuolar littering exporter that we were looking for. And as a first pass, when expressed in our tropane-producing yeast strains, 2 of the 3 transporter candidates, POP1 and LP1, increased accumulation of hyoscyamine and scopolamine. So this result suggested that these 2 transporter candidates may potentially have transport activity on molecules in our tropane pathway. So the next question is, where might these transporters be acting? Because obviously, as I mentioned, we're looking for a vacuolar littering exporter. Microscopy studies indicated that both of these two transporters are found in the vacuole membrane, in both yeast on the left side and in native nightshade plants like tobacco on the right side. Although in both cases we saw that in addition to being localized to the vacuole membrane, there was also a smaller fraction of the transporters that localized to the plasma membrane, that's the cell membrane, which would actually prove very useful for characterizing them, as I'll show on the next slide. Now, based on the already known orientation of other characterized transporters in these families, this result suggested that both of these transporters are exporting substrates from the vacuole lumen to the cytosol, which is exactly the direction that we're hoping for. So the next question was whether these vacuolar exporters could export tropane alkaloids like littering. To test this, we used a series of alkaloid toxicity growth curve assays. The idea behind these assays is that because a fraction of both transporters is found in the plasma membrane, in addition to in the vacuole membrane, any substrates that they can export from the vacuole to the cytosol, they should also be able to import from outside the cell to the cytosol by the same mechanism, because these two transport activities are biologically equivalent. So growing yeast cells expressing these transporters in high concentrations of different alkaloids should reveal to us which substrates are transported most effectively based on the relative decrease in growth rate caused by uptake of these toxic alkaloids by the transporters. In other words, the transporters should sensitize the cells more to any substrates that they're able to actually transport. And based on these growth curves, which I'll just summarize here, we identified that neither of our two transporter candidates have any activity on tropane, that both of them, and in particular PUP1, can transport littering and very efficiently can transport hyoscyamine, and then neither of them can transport scopolamine. And so these assays showed us that both of these transport candidates were rather substrate-specific exporters for littering and hyoscyamine. And so collectively, these results confirm to us that both of these novel transporters have the missing transport activity that we were looking for, the ability to export littering from the vacuole to the cytosol, as well as we learned, to reuptake any extracellular alkaloids back into the cell. And most importantly, incorporating all of these different transporters into our engineered yeast platform enabled an over threefold improvement in our final product titers, highlighting not only the importance of transport optimization to support these sorts of spatially organized pathways, but also how existing machine learning tools like neural networks can be applied in new and interesting ways to support metabolic engineering efforts. And as the complexity of these metabolic pathways that we reconstruct in microbes continues to grow, these sorts of compartmentalization and transport strategies are going to be essential in making full use of microbes as cellular factories. And so following along this theme, in the fourth and final case study, I'll describe how some new and unexpected sources of information like cheminformatics and ecology can be applied to inform our decision-making when we're designing and expanding biosynthetic pathways. So as the title of this slide suggests, the majority of commercial tropane drugs that are on the market today are actually N-substituted derivatives of hyoscyamine or scopolamine. This means they basically have modifications on the nitrogen atom. And there seem to be two major reasons for this. First, the pyrrolinium ring of these molecules, that's the 5-membered ring that actually has the nitrogen on it, that's the part of the molecule that actually binds to acetylcholine receptors in the peripheral nervous system of animals. And so modifying that ring actually has the biggest impact on the bioactivity of these drugs. And second, N substitution, that's modifying the nitrogen, seems to also have the biggest impact on the solubility and membrane permeability of these molecules, which means it's also very important in determining their bioavailability, their pharmacodynamics, and their toxicity. So for all these reasons, as a sort of proof of concept experiment, we tried to make some new N-substituted tropane derivatives using our engineered yeast platform. In order to identify potential tropane derivatives, we used a very recently reported computational toolkit called Atlas X that was developed by our collaborators in the Hatzimanikadis lab at EPFL in Switzerland. And I highly encourage any researchers who are interested in exploring metabolic pathway expansion or just exploring the derivative space of an existing pathway to check out these tools. So using this toolkit, we generated a map of all of the tropane derivatives that can be made in one biosynthetic step from hyoscyamine and scopolamine using all of the known enzyme activities that exist in nature. And from this map, we identified one specific class of N-substituted tropane derivatives that might be feasible for us to make in yeast, N-oxygenated tropane N oxides, which are shown here on the left. They all have this extra oxyanion hanging off the nitrogen. So we set out to try to discover an enzyme that might be able to perform this N-oxygenation reaction. But this is obviously a very different kind of enzyme discovery task from what I showed previously, because here, not only are we looking for a missing enzyme with a desired activity, but we also don't actually even know if such an enzyme even exists in nature. So to address this very unique type of discovery task, we again used another tool developed very recently by our colleagues at EPFL. And this tool called Bridget, which is part of this AtlasX toolkit, uses information coded in the geometry of enzyme active sites and the atom connectivity of substrate molecules in order to predict which already known enzymes in nature are most likely to catalyze a brand new reaction. So given the N-oxygenation of hyoscyamine and scopolamine as inputs, which are shown on the top right of the slide, this program called Bridget predicted an enzyme called senescyanine N-oxygenase, or SNO, as the most likely candidate enzyme to catalyze tropane N-oxygenation. This SNO enzyme and its close relative, also called PNO, evolved in insects in order to detoxify plant alkaloids found in the diets of these insects. Now, this is where, at least in my opinion, it gets very interesting, because the substrate specificity of these N-oxygenase enzymes seems to have co-evolved along with the dietary diversity of their insect hosts, meaning that as the insects evolved to feed on more and more plants, their N-oxygenases also evolved to accept more and more alkaloid substrates. So because of this, we selected orthologs from 3 different insects in order to test in our yeast platform. First, TJSNO in the bottom left from the cinnabar moth, which is a specialist herbivore that feeds exclusively on one species of wildflower and is therefore expected to have an N-oxygenase enzyme with a very narrow tolerance for only a single alkaloid substrate. GGPNO from the Nevada tiger moth, which is shown in the bottom middle, which is a more generalist herbivore that feeds on a wider variety of wildflowers. And finally, ZVPNO from the Congo harlequin locust, which is a pervasive agricultural pest, feeds on any and all plants it can find, and is therefore expected to have an N-oxygenase enzyme with by far the widest tolerance for diverse dietary alkaloids. So testing these 3 insect enzymes in our yeast factory, we saw that, as predicted from the insect's dietary ecology, it was only the locust enzyme that was able to successfully convert tropane alkaloids into N-oxides in yeast. Now, this was a really fascinating result, not only because we are now able to make a brand new class of tropane derivatives in yeast, but also it's really rare for ecological information like insect diets to be relevant for a yeast metabolic engineering project. And so this result really showed us the value of combining traditional information sources like computational tools for chemistry with, I guess you could call them non-traditional information sources like ecological relationships when you're selecting enzymes to use in engineered pathways. And it's kind of made me curious if all this time as a field, we've been ignoring all these ecological parameters that might have informed pathway design and enzyme selection. So throughout this talk, I've sort of alluded to this paradigm shift that seems to be taking place in metabolic engineering. First, a shift away from using laborious traditional genetic tools in plants and towards using more accessible public sequencing databases and microbial screening platforms for rapid pathway discovery. Second, a shift away from thinking of cells simply as bags of enzymes and towards a more integrated and spatially organized cellular factory view with carefully optimized protein trafficking pathways, cellular machinery, compartmentalization, and sophisticated transport systems, just like you'd find in an actual factory. And third, a shift away from relying solely on traditional sources of enzymes and metabolic information and towards a more multidisciplinary approach that combines bioinformatics, chemoinformatics, organic chemistry, and ecology to make informed decisions about pathway design and enzyme selection. And of course, this shift, as I mentioned, is being driven and enabled by rapid advances in both whole genome or transcriptome sequencing, as well as by rapid advances in gene synthesis. So I hope I've been able to share or confer with you a little bit of the excitement that I felt when I think about the kinds of paradigm shifts that are coming next in this field as gene synthesis and gene sequencing continue to advance at this rapid pace. But I should note at this point that there is still a long way to go from the type of proof of concept microbial platform I've described in the examples today to actually seeing yeast produce medicines on drugstore shelves. But there are significant advantages to making this transition in supply chain. By our estimates, for every dose of these drugs, a scaled yeast fermentation process will take up about 1/10 the land area compared to a medicinal crop plantation, not to mention significantly less water and fertilizer use, less material transport requirements, which means lower production costs, and overall a much smaller environmental footprint. And a fermentation process means that you'll lose much less of your product to unforeseeable events like weather, climate change, pest infestations or natural disasters. And overall, these factors mean that drugs are cheaper for countries to produce and easier for people around the world to have access to, while also reducing the chances of unforeseen drug shortages like we faced during the pandemic. But it's also worth mentioning that the adoption of this technology might also affect geopolitical disparities in different countries' access to infrastructure. Because while plantations might be costlier in the long run, it doesn't take a lot of advanced equipment to grow a plantation of trees, and certainly compared to setting up a yeast fermentation plant. And so countries with limited access to advanced chemical production facilities might be left even further behind with the global adoption of this fermentation technology and with the transition in labor from plantations to fermentation. And so clearly a very careful analysis of the socioeconomic impacts of this technology will be very important moving forward. So with that, I will conclude by acknowledging the very important contributions of a whole team of people over the course of my PhD to the success of this yeast factory project, including Stanford professors Christina Smolke and Elizabeth Sattley. Several members of all of these labs are tireless support staff and our collaborators, and of course my funding sources. And at this time, I would be very happy to take any questions. Thank you.
Thank you, Prashanth, for that fantastic presentation. We're now going to open it up the floor for questions. If you have any questions, please make sure to ask in the Q&A box. I see that we have a couple of questions. The first one from Nazca. Thank you for sharing your interesting work. In your opinion, what subtypes or types of nightshades are most valuable in drug discovery and why?
Yeah, that's a great question. So I might be a little biased because I like the whole nightshade family myself. I grow all those plants myself. I think they're all very valuable plants. From a medicinal perspective, obviously, nightshades that produce these kinds of tropane alkaloids are really absolutely critical. When I really first started this project, I had no idea how important all of these drugs were to modern medicine. And obviously, any plants that produce them are not only in kind of short supply now, but are also a vital resource for discovering not just the biosynthetic pathways for these drugs, but any important pharmaceutical derivatives that we might be able to make from these compounds. But at the same time, I also have to mention that even nightshade plants that don't produce any tropane alkaloids are still a valuable resource. I mentioned how tobacco, which is also a nightshade plant, is used as a sort of model organism because it has, it shares a lot of the same genetics with tropane-producing nightshades. So it's a very valuable host for troubleshooting protein and enzyme expression and gene expression when things go wrong in yeast. And so, I'd say that even non-tropane-producing nightshades are still very important. As far as which subtypes that might be most kind of pharmaceutically relevant, at this time, Duboisia, which is native to Australia and Oceania, is probably by far the most pharmaceutically relevant just because that's the industrially farmed plant that produces these drugs. A lot of the other species that I mentioned, deadly nightshade, Tropa belladonna, Datura, Bergmansia, these are all grown, more in the wild or even as ornamentals, but they're not really conducive to mass production. And so Duboisia is really, because it grows more as a tree, is very space efficient, at least compared to those other plants, is probably the most important kind of subtype and clade for pharmaceutical purposes. I hope that answered your question.
Okay. Thank you. We have a few more questions. Neil asks, is the final product secreted? And does purification from the yeast process pose any challenges or limitations?
Yeah, that's a great question. So that's one question we always have to be kind of concerned about anytime you build a microbial process. Really, anytime you're producing a valuable compound in a living organism, whether it's plants or microbes. In the case of plants, the supply chain basically just relies on harvesting the leaves, drying them, and there's an extraction process. In yeast, we try to build our process as much as possible to secrete these molecules into the media. It's orders of magnitude easier to build an industrial fermentation process where you just have to extract molecules from the spent fermentation broth than if you have to actually crack your yeast cells open and extract it from there, because performing yeast lysis on an industrial scale is just really a nightmare. In the case of the tropane pathway, we're reasonably fortunate that a lot of the compounds we're making in yeast are actually toxic, and so they're naturally secreted from the cell in an effort to sort of detoxify themselves. So all the experiments we did and all the kind of process development all kind of depends on analyzing production in the extracellular medium, in the supernatant, once we've kind of centrifuged and pelleted our cells. But as I mentioned, transport is an important kind of consideration, and we know from past experiments that a lot of these alkaloids can hang around inside the cells, and they have kind of different residence times inside the cells. And so optimizing the secretion of these molecules to the media is a very important part of the sort of scale-up process. And so that's just one of the ways that yeast engineering has to be kind of implemented to promote a type of fermentation process that's kind of more conducive to an industrial supply chain.
Thank you. Stephen asks, what are the advantages to using yeast as compared to bacteria?
Yeah, also a very important question we ask at the beginning of really every metabolic engineering project, because you're always kind of faced with this choice of, do we use E. coli or some other bacteria? Do we use yeast or some other type of fungus, eukaryote? I will say, I mean, E. coli have a ton of advantages. They grow much faster than yeast. They're just generally kind of more easier to culture. They also, talking to the previous audience member's question, you have a lot fewer transport issues with E. coli because they don't have compartments and their cell membrane is a lot more permeable. So you don't have to worry as much about molecules being trapped in the cell. But that same kind of simplicity also comes with its own kind of disadvantages, especially when you're working with plant biosynthetic pathways. So plants, as I mentioned, have sort of compartmentalized their pathways with different portions of the pathway taking place in the chloroplast, mitochondria, ER, vacuole, et cetera. And if you were to move this type of pathway into E. coli, a lot of those compartmentalizations just kind of fall apart. I'll give you as an example one very common failure point that we see, which is working with cytochromes P450. These are enzymes that are typically tethered in the phospholipid membrane of the endoplasmic reticulum. They need a sort of membrane anchor in order to kind of function. And so when you move that to E. coli, obviously E. coli don't have an ER, so you either have to engineer the protein to try to make it into a sort of soluble form, which is very hit or miss, or you have to kind of rely on trying to force it into the plasma membrane, which again also comes with a host of issues. So generally for plant pathways, it's most convenient to use a host that already has a sort of cellular architecture that matches the original host, which is plants. But yeast also have a lot of other advantages. I mean, we have very well-developed processes for kind of building fermentation processes around yeast. We've been working with yeast for millennia as humans, and certainly at an industrial level for over a century. And so there are very well-established protocols for scaling yeast processes up from lab scale to pilot scale to industrial scale. Yeast are also a lot more robust than E. coli. They can take a lot more punishment basically before they die, which is again also very important at an industrial level where you can't always be as careful with maintaining the perfect conditions for growth. And just in general, in my own personal experience, I've found that yeast are a lot easier to engineer at a genetic level than E. coli. I know that might be kind of surprising given that E. coli are just prokaryotic, but a lot of the more complex genomic operations that we have to do, I've just had much better success with in yeast than in E. coli. So I think for all of these reasons, I would just prefer yeast, especially as a host for any pathways that are coming from a eukaryote like plants or fungi. But obviously, each project you have to kind of consider pros and cons whenever you're making the choice of which host organism to use. Okay.
In the interest of time, let's answer a couple more questions. Yogendra asks, great work. I agree. Just curious, prior to searching for transport channels within the yeast compartments, were you able to observe the TA reaction completion by simply lysing the yeast, either chemically or mechanically?
Yeah, great question. So we tried yeast lysis as part of our troubleshooting process. I mentioned all those kind of troubleshooting steps we went through in trying to figure out why the Lutherine synthase wasn't working. And actually, initially, we were able to see our final products outside the cell, even without any of the transport optimization. It's just that they were at very low levels. As I mentioned, a lot of the intermediates and molecules in this pathway are quite toxic to yeast, and so they kind of have a natural tendency to try to excrete them from compartments and from the cell. And that sort of natural excretion tendency was what we were able to use to do the initial troubleshooting and detect these molecules outside the cell in the first place. So we were actually able to observe the TA reactions even without lysing the cells, but especially once we lyse the cells. It's just that once we put in these transporter proteins, these channel proteins, we saw large improvements in our final product and consumption of some of these intermediates outside the vacuole, which told us that transport was a major bottleneck. But yeah, yeast lysis is definitely an important tool when we were troubleshooting a lot of these reactions.
Okay, thank you.
Let's make this our last question. Have you calculated what is the approximate cost to make a wanted protein model?
Okay, so I assume by the protein model, maybe you're talking about the kind of overall process. Maybe could you maybe just clarify what you mean by protein model? Do you mean basically the kind of cost for the overall fermentation-based process? Yeah. So yeah, unfortunately, I can't talk too much about economic considerations just because this is ongoing industrial work. But we did do some initial calculations when we first published the overall pathway in yeast as a sort of proof of concept. We did some calculations just to kind of determine, just to self-check if a fermentation process based around this would be more economical than growing plants. And yeah, I mean, I kind of showed that on one of the last slides. One of the major improvements in cost in this process is basically going from growing, the original medicinal crops on a plantation to growing sugarcane, which is essentially that becomes the sort of plant input to the process. Instead of medicinal plants, you just get sugar from either beets or sugarcane, et cetera. And the cost estimates for growing sugarcane versus growing medicinal crops is just orders of magnitude lower, because we have very well established protocols for for growing sugarcane and sugar beets, and there's a lot more kind of care you have to take in growing medicinal crops in terms of making sure that the conditions are perfect to promote alkaloid production, promote growth conditions that promote pathway activity. Because one of the first things that plants do is sort of kind of shut down a lot of these secondary metabolism if they're under conditions where survival is threatened. Unfortunately, I can't give you specific numbers, but I will say that it was about an order of magnitude drop in overall cost. That includes, resources, time, land area, et cetera, going from a medicinal kind of crop supply chain to going from, sugar from sugarcane into a yeast fermentation process. Hopefully that was able to answer your question.
Thank you, everyone. I see that we do have a few more questions. Like I said, we will give these questions to Prashanth, and we have your email addresses, so we will respond via email. With that said, just a quick reminder that following this webinar, you will be redirected to a brief survey. If you have 1 minute to spare, we would appreciate your feedback. And finally, one last time, let's thank our speaker for a fantastic presentation. And of course, thank you everyone for coming, and have a wonderful day.